morning, everybody. Thank you for coming to this very popular symposium. First, I would like to thank all the organizers for this meeting, in particular Dr. Carol Reif, Teresa Berry, and Todd Forsyth for doing a wonderful job in organizing this meeting. So I see that most of you are aware of the things that you can do to promote healthy aging and healthy cognitive aging. And what I'm going to talk about today is the things you don't have control over, or you might, as I hope to show you today, and that's the genes. So my lab is interested in finding out the genetics of learning and memory and aging. And we use rats as a model because, like humans, well, first of all, they're very smart. And like humans, when they get old, you have some rats that can remember as good as like young rats. And then you have all the other side of the spectrum to rats that are completely learning impaired. So they're a very good model for the variability that we see in humans. And so what I hypothesized before I came to Madison was that these differences in cognitive ability in these old rats were due to differences in gene expression. And what do I mean by that? So I think because there's two screens, I'm going to use this instead of the pointer. Does it show? I guess I'll use the pointer. I'll have to choose one side. Sorry for you guys. But anyway, so these are, I'm going to talk about the central dogma of biology. And these two guys are James Watson and Francis Crick. And I think they're the most famous Nobel Prize awardees in the 20th century for the discovery that DNA structure is a double helix. So I'm going to tell you what I mean by gene expression. So we all have, our genes are coded in DNA, and they're kind of there. And they're either good, they're bad, they're mutated, we're missing a chunk, but they're there, they're static. And so then when we need to have our tissue function or a cell function, we need to make proteins, which are the building blocks of our bodies. And in order to do that, so depending on how much protein you make, you need to make messenger RNA. So you get the information out of the DNA into messenger RNA. So it's like making photocopies. So for people like you, I need to make a bunch of photocopies, like 600. So each of you has a copy of these things, and I can make tons of it. I might need less, like I might need only 20. So that's what RNA is for, is to make as many copies as you need for a certain function. And then in turns that gets translated into protein, which is the building blocks of our cells, our neurons, our tissues, like our lungs, our heart, our brains. And this process is very regulated. So for example, wound healing, you cut yourself, and so you need to make proteins that replace the skin, replace the blood vessels. Once your wound is healed, then the process stops. So you don't make any more RNA, you don't make any more protein, you're done. Now, sometimes making a lot of protein is good, sometimes making a lot of protein is bad. And this is where you have disease. And also when you have like a mutation, or like I said, there's a deletion in one of your genes, then you make defective protein, and that's what causes disease. So this is all these things can be measured, they can be quantified, and this is an approach that I use in order to interrogate the entire genome of rats to find out which genes are turned on and off when an animal learns versus an animal that cannot learn. So we use some behavioral tasks for learning and memory, and I'll talk about it in a little bit later. One of them is the Morris water maze, where we place the rats in water and they have to find a platform. And then what I did in this experiment to find out the genes is I look at the hippocampus, which is a part of the brain that is involved in the acquisition of spatial and episodic memory. Spatial memory is the memory to find out where you are, and as you know, like people with Alzheimer's have a problem with that. That's one of the first things to go. And the other one is episodic memory, and that's the memory of events. And those are acquired in the hippocampus. And it's called hippocampus. All my students are like, all these names in the parts of the brain, striatum, hippocampus, substantia nigra. And it's like, well, if you knew a little bit of Latin or Greek, you know that they all make sense. And so the hippocampus is called like that. This is a hippocampus dissected out of a brain. It looks like a seahorse. And hippocampus is Greek for seahorse. So, the anatomists in the past centuries, they were very clever, and they named things by things they reminded them of. So anyway, we train these animals, we separate them into learning impaired, learning unimpaired, and then we isolate the messenger RNA that I told you is like the photocopies of our genes to make lots of protein. And then we need to label them, like put some kind of tags so then we can quantify that. And so we do that using these microchips. These microchips, they're the size of a domino, and they contain the entire rat genome. So you can interrogate the entire genome with this labeled RNA, say which of the 30,000 genes are turned on and off when these animals can learn versus the animals that cannot do that. So when I did that, I just focused in three parts of the hippocampus, but anyway, the part that I focused on was called the cornus ammonis 1, area 1, and that's an area that's particularly vulnerable to aging and Alzheimer's. And when I did that, I found 50 genes that were either up and down in animals that could learn versus the ones that couldn't. And today I'm just going to talk about one particular one. And before I do that, I need to introduce you to the concept of the synapse. This is another Nobel Prize winner. He's one of my countrymen from Spain, Santiago Ramón y Cajal. He won the Nobel Prize for discovering the synapse. Before his time, people thought the nervous system was like a reticular system, like a cobweb. And he discovered that there were individual neurons, and they were connected to each other by a little space that's called the synapse. And that's where all the action takes place in order to transmit information from like the top of your head to your little toe so you can wiggle it. And so this is how it works. So basically, the transmission of information in the nerve system is both electrical and chemical. So first you have like this electrical signal that gets passed on via these axons, which is these long terminals, and it gets all the way to the synapse. So this is what an enlarged synapse looks like. So when you get this electrical signal passing on, this cell releases neurotransmitters. Neurotransmitters are either amino acids or peptides, and what they do is they bind to the next neuron in line, and they send this information. They bind these proteins in their surface, and then these in turn then can cause this next neuron in line to fire and transmit the information, and so on and so forth, from the neuron that's connected all the way to all the neurons in the tip of your toe, Anyway, so now I'm going to talk about my gene of interest, which is HOMR, which is actually located in this part of the synapse. HOMR1 was one of the genes that we found in this chip interrogation assay, in this gene-wide expression assay. And it's a protein that is expressed in this side of the synapse. So basically, it receives the information from the incoming neuron, and then it activates and triggers a cascade of events that results in the activation of this neuron, and eventually leads to learning and memory, which I'll talk in a little minute. And what we found is that one particular form of HOMR, which is HOMR1C, the messenger RNA was down in animals that could not learn. So the first thing we do in genetics when we try to show that a gene is important in a given function, in this case learning and memory, is we knock it out. We basically take it out of our genome. And then we see if these animals are stupid, right? If you remove a learning and memory gene, then they'll be dumb. And then they'll prove that, this is a gene involved in learning and memory. And so actually that experiment was done. There's this Peter Seberg and Paul Worley created a mouse model where this gene was, we call it knockout, and sure enough, these animals show deficits in learning in the hippocampus, so learning of spatial memory and episodic memory. And we know that our age rats, they also have hippocampal deficits. So then we were thinking, well, so these animals, we know they have low levels of HOMR1C. What if we put it back? Can we restore the learning in these impaired animals? Or in other words, can we make smart rats? And the answer is yes, we can. And I'm going to show you how. So I use gene therapy approaches to treat neurological disorders, but also to ask questions about learning and memory. So I'm going to describe what gene therapy is, and basically it's the use of DNA as a drug. To correct, to deliver into a cell and to correct a defect. And so the DNA can be different forms. It could be a therapeutic gene. So for example, people who have hemophilia, they have a mutation or they lack the clotting factor 9, which is a protein that is involved in clotting. And so people have actually replaced that gene into the bloodstream of these patients. And now these people that used to need blood transfusion on a weekly basis, they're done. They just don't need it anymore. So it's a pretty impressive, successful story for gene therapy. Another way to do it is just removing the genes that are mutated and are toxic. So in the case of Huntington's disease, the gene huntingtin is very toxic, and so people have found ways to kind of cut up into pieces that mRNA and remove the toxicity. And the way gene therapy works is by delivering your genes using viral vectors. These are viruses that have been engineered to remove all the bad genes that the virus has and replace them with a therapeutic gene. Why use viruses? Well, because they have evolved over millions of years to do exactly that, to deliver genetic material to the cell, and they use the cell machinery to make their own proteins. In our case, it's going to be our own therapeutic protein. So basically, you have here a virus, and it gets into the cell, it goes all the way to the nucleus, delivers the DNA, then the cell machinery, so it kind of scavenges the cell machinery, makes RNA, makes protein, and then there you go. You have your corrected protein. So in order to get them into the brain is kind of tricky, and so we use these very minimally invasive and highly accurate injections. Yeah, you laugh, but I've seen what neurosurgeons do, and they're not much different. I mean, they're more like the Black & Decker. I mean, they have power, you don't have to do, but... So anyway, we have maps of the brain that very accurately tell you where each part is. And so we can target the hippocampus and just deliver the gene into just that particular region. And so this is how basically it would work. So we would have our virus that codes for Homer, and this thing would be the protein. So we have the brain, and then we target that injection site into the area we want. The virus is the liver. We actually call them viral vectors because the viruses are bad and the viral vectors are good. And then the virus does its thing and starts making the protein, in this case, Homer. And so that's what we do, what we did like in our age-impaired animals that I'm going to show you in the next study. So I told you that we have some age rats that are impaired and others that aren't impaired. So our first experiment was to see if we could rescue the deficits in these animals. So in order to do that, first we have to take all the rats because some are smart, some are dumb, some are in the middle. And so we do a behavioral task that I'll show you in a minute, and that's called the object location memory task. And then we separate them into learning impaired, learning unimpaired. And so then we take the learning impaired rats And then we inject half of them in the hippocampus with our Homer1C gene, or as a control, you always have to use a placebo control, although animals don't know what you're doing to them, for green fluorescent protein, which is an innocuous protein that glows in the dark, which is nice to then see where you inject it. And so we call it like GFP. And then we wait for a few weeks so the virus does its thing. It gets into the cell, turns DNA into RNA, RNA into protein. The protein does its thing, and the animals get smart. And then we test them again. And so we test them again in this object location memory task, and then we do an additional task just to double check that they're smart in different tasks. And this is the Morris-Water maze. And so, okay, so here's the first task. We place the rats in an arena that has two identical objects, usually like, little construction blocks. And the rats are very curious. They like to explore, and so they're going to explore both objects. And eventually they figure out that both objects are identical, so they'll spend 50% of the time in one versus the other. Then the next day, we move one of the objects to a different location. And so if the animal has an intact hippocampus, which is important for spatial memory, like I told you, he'll say, hey, you move the stuff. And so he'll spend a significant amount of time exploring the novel location. If the animal is impaired, he won't even notice the difference. And why do they care that we move the object? Well, as you know, rodents, they bury their nuts and they need to find where they place them. So they need to remember where they were. So this type of behavior is very important to them. The second task we do is called the Morris-Water maze. And this was developed by Richard Morris, and is based on the fact that actually a lot of people might not know this, but rats are aquatic animals. They love to swim. And I actually seen them swim in the Yahara River when I walk my dogs, and probably you don't want to know this, but they're taking their, daily baths. And, but anyway, so what we do in this task is we have here, I don't know if you can appreciate it, in one quadrant, we have a hidden platform. It's submerged a little bit under the water, so the animals cannot see it. And they basically have to use spatial cues in the room, again, spatial memory, to find out where that platform is. So we drop the animals at different quadrants, and then they start swimming around, and they're like, whoops, they find it, and then they climb on it, and they like look around, and they're like east, north, west, south. Of course, if they don't find it, we put them back there. We don't wait until they drown, right? And so we put them there, and then they get their bearings, and we repeat that like about 4 times every day, and then we do it over a number of days. And so eventually the animals start circling around. Wherever you drop them, they're like, oh, it's right there, boom. And then the distance to find it, it becomes shorter and shorter. Then we play a nasty trick on them. Then we remove the platform because we want to test if they really learn what the platform is. So if the animal has learned, he has an intact hippocampus, when we put them there, he'll start circling like, I was pretty sure it was here. And then, and so we count how many times the animal crosses the space where the platform was. And so if it hasn't learned, then he'll circle around and spend a random amount of times all over the place. Okay, so, whoops, I'm not supposed to show this. Yeah, it's a very busy slide, so I want to do it in parts. Okay, so I'm going to explain. This is the novel object location test, the results. In the y-axis, I'm showing the novelty index. That's the percent of time that the animals spend exploring the novel location. So 50%, this line here means they're spending 50-50, so they really haven't noticed that we have moved the object. Above 50 means they are expending a significant amount of time exploring the novel location. So they have figured out that we move the platform. They have an intact hippocampus. On the x-axis, I have the 3 groups. AI, which is learning impaired, age impaired, injected with Homer1C. Then we have the age impaired injected with a controlled GFP, and then we have the young. And we always use young as a reference for awesomeness. It's like, they're so good at learning. We always need a reference point. And so when we do that, and then OLM1 is, we do the test before before treatment. OLM2 is after we've delivered the gene of interest. So what we see, again, in the first two blocks, what we have is the animals in the beginning, they were performing at around 50%, but after we delivered HOMA1C, they significantly improved. And you can compare them to young, they're doing as well as young rats. On the other hand, the animals that were injected with GFP before, after the treatment, they don't do any better. And also some of them actually do worse, but that's because they're getting older. We wait a few weeks and it doesn't get any better. So this was very impressive, and we concluded from these results, and we also done that in the knockout mice, that delivery of HOMA1C into the hippocampus of age learning impaired rats significantly improves cognitive ability. And we also saw some improvement in the Morris-Water maze that I'm not showing for the sake of time. So then we're like, okay, this is great, but what's happening in the neuron? I mean, that's what we want to know. And so in order to do that, we decided to do a different approach because, gene therapy is great, but I mean, how many of you would like to have a whole drill to become smarter? Yeah. So my grad student at the time, yeah, me too, but my grad student at the time, she said, this is great, but why don't we try something a little less invasive? So why about environmental enrichment? And so environmental enrichment is being shown both in humans and animals to improve cognitive ability. And the thing is that we just don't understand why. I mean, we know that people with high cognitive function, they have resilience and their resistance to the effects of cognitive aging and Alzheimer's. But we don't understand the mechanism. We don't know what happens in the brain. And that's what we are trying to find out. So for this, usually when you have rats in a lab, you have them in a cage that holds 2 rats. But in this case, for enrichment, we use a big cage that can hold 6 rats. And then we have igloos, and then we have nesting material, and then we have pipes, and then we have toys that we change every week. So there's novelty. And so these animals basically, they have both intellectual enrichment, and they also have a bunch of friends, and so that's social enrichment. And, we want to know which one of those is important. Do you need to have both? And so in order to find out if social enrichment would be enough, we had another group of rats, and these rats, they also have a bunch of bodies, but no toys. So, it's just hanging out with friends. And then finally, our control rats are the rats, our typical cages where we normally house the rats. So comparing this to what human enrichment would be like, so environmental enrichment, as we call it, would be, like you guys here with your friends, but learning something new, versus social enrichment, which is just hanging out with your friends at the bar or something. And then our control would be just couch potatoes. Okay, so we had animals housed in these 3 different conditions, and so then we tested them in the Morris-Water maze and in the novel object recognition test. So what we find, which is what other people have found with enrichment, is that you get improvement in learning. So this is the total distance. The y-axis on A is the total distance to find the platform. And on the top, the animals that are the couch potatoes, the standard cage, they don't get any better with training. Whereas if you look at the bottom graphs, both the environmentally enriched and the socially enriched, they get better. When we do remove the platform to see if they really have learned where the platform is, we find that actually the animals that are under environmental enrichment perform significantly better than both socially enriched and the cage controls. And similarly, when we do the novel object recognition task, what we find is that the environmentally enriched animals spend significantly more amount of time exploring the novel object than both socially enriched and environmentally enriched. Okay, okay, so great. So environmentally enrichment works, but again, what's going on at the cellular level? And so I'm going to show you how we think memories are formed in our neurons, what we think happens. So this is the hippocampus of a rat that we dissect out, and we can actually record information for 12 hours after we put them in a little dish. And which is, I think, kind of impressive, a little Frankensteinish, but it works. And so here what we're trying to do is look at these synapses that I mentioned before. So we have this neuron here that's firing, and it's releasing these neurotransmitters to the next neuron in line, which is our neuron of interest. That's where Homer is expressed. So basically what we do is we have a stimulating electrode in this area where these neurons are, and then we record and look for the response of these neurons over here. And so this is how it works. You basically start sending small impulses, so you can check that your hippocampus is alive. And once you establish a baseline, you see the cells are stable, they're not dying. Then you hit the neurons with high frequency stimulation. And usually there's a number of paradigms to do this, but one common one is called theta burst stimulation, and it mimics the wave frequency that the hippocampus produces when you're learning something new. So basically you go boom, you hit it once, you hit it twice, you hit it 3 times. So now what happens is, I'm showing here an action potential, which you've probably seen when they put you on an electrocardiogram. So you see the firing of the heart cells. This is similar with the neurons. And then when we do this, then actually, boom, these neurons start firing much higher rates. And what we measure is the difference in the slope between the black line and the red line that is much steeper. And that's what's expressed in the blue graph here. Now, you would expect that after you're done with hitting them with high stimulation and getting this high response, that then the neurons would go back to baseline, right? Because you're done. But the neurons remember that they receive this kind of information. So this is what's happening in your neurons when you're acquiring new information, and then when you get that information again, you're like, so this is your connections are potentiated. So we call it long-term potentiation. And actually what people have looked at the anatomical level is that these synapses get fatter and more stable. Usually these synapses kind of come and go, but when this happens, it's like, I'm gonna stay, and this is a memory, okay? So then we use a different approach because I haven't mentioned, but Homer interacts with a receptor in the membrane that's called metabotropic glutamate receptor, which I'm going to call mGluR to make it easy. Basically, so I told you, if you give a strong stimulus, you get this long-term potentiation. But if you give a small one, you have this decaying wave. So, but if you add a pharmacological drug that activates these mGluRs, then you can convert this short-term potentiation into long-term potentiation. So basically, in this synapse, and like I said, this synapse is getting fatter. So when you add this pharmacological agent, you're releasing in here, and you're strengthening the signal. So this is the type of testing we do to see if our animals are smarter or not. So when we did this in standard animals or in animals that are socially enriched, what we find is that you need this drug to enhance long-term potentiation. So then we're like, okay, let's do it on the environmentally enriched. I would expect that then this would be like much higher, like right here, right? Well, no, actually, these guys don't need the drug. They're already primed. So just this little burst causes the cells to remember. So that's what is reflected. That's why they're smarter, because they need less information to carry on. And so if we add the drug, they don't get any better. They're already maxed out. So they're like super smart. They don't need any kind of drugs. Some of us do, but. And so in conclusion, we have found a gene that's called HOMER1 that is important for learning and memory formation, and that this gene is expressed at low levels in 8 rats that are learning impaired. And then when we put that gene back into these learning impaired animals, we find that we can rescue the learning deficits. And one thing that actually I haven't put in the slide that I forgot is that I've shown you these data is animals that were enriched. People have done a lot of enrichment where they have tried different lengths of time. Do you need to be intellectually stimulated from the time you're young? Is it good enough if you start when you're old? And there's been many studies that, do you need enrichment all day long? Do you need enrichment a couple of hours a day? These animals, so we've shown that this mechanism occurs both in young and old. We started with the young animals. The animals, we have to enrich them for 4 months to get these differences in learning ability. But then we took 2-year-old rats, which are very, very old rats. They're about to, go. And with only 1 month of enrichment, they were able to change their anatomy of the brain and their function. So it's never too late. That's the take-home message for that. And then, so basically what we are starting to understand is that environmental enrichment enhances this form of synaptic plasticity that we call long-term potentiation. And this enhancement is dependent of HOMER and also this other protein that's mGluR5. So now we're trying to go and say, well, what's happening at the molecular level? What are these 2 proteins doing? And so this is some active research in my lab right now. One thing we know is that I have a folder that's called Damn Signaling Cascades, because I have bad memory. That's what I do this. I'm trying to cure myself. And so to remember all these names is kind of like torture for me. But anyway, so don't pay attention to all the names. Basically, what the take-home message is that this metabotropic glutamate receptor interacts with HOMER, and they both need to interact to start these signaling cascades. And basically, they activate proteins, and usually they're proteins that phosphorylate other proteins, and phosphorylation is a way to kind of activate, wake up proteins. And eventually, all these cascades lead to the translation of proteins in the part, in this synapse region that make the synapse stronger and are important for memory and plasticity. And more importantly, what we found with the enriched animals is that these P70S6 kinase, which it's actually activated for longer time than in control animals. And so we think this is the key of why these animals are smarter because of this protein. But curiously, this protein is also constitutively activated in Alzheimer's disease. So too much of a good thing can be bad. And so that's our next area of research, is finding out why in environmentally enriched animals, this thing can turn on and off, and in Alzheimer's it cannot. And so that's basically where we're at as I speak. And I just want to acknowledge everybody that work in what I've presented, which was Sue Osting is my lab manager and surgeon extraordinaire. Hilary Gerstein did the work with the HOMER knockouts animals that I didn't have to present, but actually she won the New Investigator Award in 2010 here at the Institute of Aging for the work she did. Then Ken O'Riordan was a technician in my lab who now got his PhD in Ireland. Ricky Hollinger, she's also got her PhD, and she's the one who did all the enrichment experiments. And then Mary Listrom is a statistician who helped us navigate through the statistics of all these behavioral tasks. And this is the funding. Any questions?